This is Professor Alan Haungs, Physics 124, Lab 10, Lenses, Mirrors, and Optical Instruments. Um, for this lab, I thought it would be interesting that since many of you are biology students, that you learn what is a confocal microscope, what is confocal microscopy, why does it exist, and how does it work? Um, You're looking at the original patent by Mr. Minsky in 1961 when he submitted his patent for the design. He was way ahead of his time. You can get the patents, you can get all that stuff online right now. So I'll explain it to you. First, I'll put a little background, and then I'll explain the physics of it. And then you can do a little lab experiment with the computer software, and just to kind of prove to yourself how it works. What are you looking at right now? Can you tell me, what are you looking at right now? From this view, can you tell? Maybe. What if I go like this? Oh, it's a bowl. It's a bowl in a bowl. A bowl inside of a bowl. That's all it is. Okay? So without some kind of three-dimensional ability to discern something, you may not really have a good idea of its structure. So if this were a cell, you would have a problem in the old days when you take a cell and you put it on a microscope slide, and normally you're looking at it from the top down like this. And you don't get much three-dimensional perspective at all. The other thing is that, the parts of the cell are both transparent and cloudy at the same time. So there might be something at the bottom that you want to take a good look at, but you can't because of all the material on the top is cloudy. And then you're not sure when you're looking at it like this, what part's the top, what part's the bottom. Are some of these parts separate? Is this part separate from this part? Are these two parts or one part? Or, you wouldn't be able to tell in the old days with a microscope. What they used to do is they would defocus the microscope and try to focus it at different depths. Start here and go deeper and deeper. And that would help you get an idea. So this guy named Minsky, he had a great idea. He essentially he envisioned a concept in which you could scan. Just imagine if I could do this. If I could scan across the tip of my pen here would be the only spot that I could see. It would be an infinitely small spot. And I would just scan, and I'd look for light and dark, and then scan again, light and dark, and then scan again. And eventually what I would do is I would make these scanning motions across the whole surface at this depth, right? If I could do it at only this depth, right? Or if I could do that same scanning motion at the depth at the halfway point, and I could only scan the light emanating from that sheet, from that plane right here. Is that possible to do that? It is possible. He invented a way of doing that. Now, if you can scan and only see the light that's coming from this level, essentially you've made the top invisible, and then the bottom isn't clouded either. It's not emitting light. So it's all as if you made everything else other than that one sheet, that one layer, visible. So imagine if you could scan a layer, scan a layer, scan a layer, and then scan a deeper layer, a deeper layer, a deeper layer, and then scan another deeper layer, point by point by point. Just like as a TV set works, right? How's a TV set work? It goes like this. It scans like this, and it goes back and scans like this, and then scans like this. This is how your TV set paints a picture. And then it scans. And then as it's scanning, it's either that's bright or that's dim, or that's bright or that's dim. So these we call those pixels, right? So you create this raster of bright, dark, bright, dark, bright, dark. So how could you do that? Well, in our earlier lab, I said that you could use a pinhole to create an infinitely deep depth of field. If you're like me and you're older and you're farsighted, if you go outside and you're under the bright sunlight and you're reading the newspaper, you may not need to wear glasses because your pupil will contract, and it'll create sort of a pinhole effect, and you'll be able to actually see the letters on the page pretty clearly. Otherwise, without the bright light, you'd need to wear a pair of reading glasses. So a pinhole can create an infinite depth of field. Here in his invention, he's using a pinhole to create an infinitely narrow depth of field. And how does it work? Well, let's just look at his drawing. Very simple. This is the one embodiment. If this is the area where the cells are, picture this as a semi-transparent medium in here. If I have a light bulb over here, and I'm reflecting the light. Now, this half of the experiment is why it wasn't very practical. Initially, when he invented this, it wasn't practical until actually lasers came along. But we'll just take his original invention and explain it. If you take this light and you only pass it through the narrowest hole, exactly at the focal point where this lens would focus light from here to here, it would perfectly focus it, you could create a perfectly fine point of illumination right here. Now, this is a very inefficient way of creating a point of light at a specific depth. Now, we use lasers to do that today, but back then, this is how he perceived the idea. This is just the exact mirror opposite of it. This is a photo detector. This lens would focus the light exactly from this spot to exactly through this narrowest of hole, and it would pass through the hole, and then it would go into a photo detector. So we're not focusing an image on this side. All we're doing is we're looking to see how bright is the light coming from that exact point right there. That's all we're looking for. And the only light that can get to this through this detector would have to follow a perfect path so that it goes right through this narrowest of hole. If this thing were slightly higher or lower in this plane, these rays would converge somewhere above this hole or somewhere below this hole, and it wouldn't get to the detector. If this light, all the light that's emanating before or after this focal point here, what it does is it then converges in such a way that it would try to converge back here. The majority of the light can't get through because only the most central part, so most of it's wasted. If it converges too early, it gets broad again, most of it doesn't get through. So you've literally limited this to only looking at a very smallest spot of light at an exact depth in an exact place. So imagine if you could then just take this sample and just move it up and down, and then also in and out like this, and then you could create a raster of the image. So that's what this next drawing here is basically. He's essentially saying if you were to look, if this is your sample right here, if you're looking down at it, and these are electromagnets, and they're essentially shaking it like this. They're shaking it like this. Bang. 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 And then he has his photo detector hooked up to an oscilloscope. The oscilloscope would retrace real quickly each of the values of the brightness of the light viewed by this apparatus above or this one below. This is just a different embodiment of the same idea, cleverly using a mirror that will either transmits or reflects light depending on the direction. So if it goes this way, it goes this from here to here. It transmits light coming back, then gets reflected down. So it's the same idea. Anyway, so what he did is he hooked this up to an oscilloscope, and as this is shaking the sample, it's then painting a picture on this oscilloscope, the bright, dark, the bright, dark. So this is what all these connections are for. These are the oscillators that drive these electromagnets. That's an oscillator right there. Hooks up to the oscilloscope. It synchronizes the scope's scan, the vertical and horizontal scan. Same thing with here. It's an oscillator, electromagnet that vibrates the whole thing. So that was the idea. So this guy was way ahead of his time. It wasn't really practical to build these until lasers came around, and then this half of it basically was replaced by lasers. But this is still the other part. So it's called a confocal because there's a shared focus by two stereo halves. The light source and then the light observing, the light sensing. So that's it in a nutshell. Well, next thing we'll go to the computer, and we'll look at a setup that I made, and then you can do some experiments and hand in a lab to me if you decide you like this lab.